Hello. This is going to be the first of 3 lectures on chapter 3, and I have put into Canvas um, the slides. There's one set of chapter 3 slides, but it's quite long and it's far too much for 1 week, too much for 2 weeks for that matter. So I've divided it into 3 weeks, and we will go through in this first lecture, slides number 1 through 14 as indicated on Canvas. Let me go ahead and share the screen, and then I'll make just a few other general comments before we get started. All right, I'm going to make that big. And okay, there we are. Yeah, so now this is Organization of the Nervous System 2. That's the name of the chapter in the book. It's kind of a mixture of things. They don't necessarily go together, but they're just pieces that ultimately you need in order to understand the neurologic diseases and injuries that we'll be studying, and how they affect communication, and also importantly, as I've mentioned a number of times already, how the different signs and symptoms that any one patient will have fit together in patterns that should match the speech language pathology diagnosis as well. So that's kind of where we're heading. We're going to go through 14 slides right now. I think it takes about a half hour, but we'll see. Of course, you can stop and come back anytime you want to. And I also did indicate the pages that this corresponds to with the book. It's a close correspondence, but it's not perfect. You'll notice some things that are in the lecture slides that actually aren't even there. Sometimes I've used some figures or illustrations that are from other sources because the book doesn't give you enough actually to go where we're trying to go. Whenever I've used a figure from the book, hopefully I've done it every single time, I indicate the figure number so that you know that does come from the book, and you could, study the corresponding areas to that to better understand the figure. But if there's no figure number, it means it came from another source. Okay, that being said, let's get started. What's going to be covered in this chapter is, it is a kind of a mishmash as I look at it. It's different things that don't necessarily relate directly to each other, but ultimately you'll need all the information. The peripheral nervous system, the autonomic nervous system. Autonomic nervous system, by the way, we're not going to study it very much at all because it doesn't have much to do directly with speech and language. It's the part of the nervous system that controls vegetative functions like digestion, for example. The place where it gets into our area of clinical study is in the area of swallowing. That there is a connection between the autonomic nervous system and swallowing in addition to the production of saliva, which is a kind of a vegetative function and is controlled by that part of the nervous system. The other thing is just the feeling that you have behind the mouth. I mean, in the mouth, the system is linked up with the rest of your body. It's basically the same system that when you touch your arm, you feel it. When you touch the inside of the mouth, you feel it. But if you go behind the mouth and into the pharyngeal area and down below, and the pharyngeal area is certainly important for swallowing, that's hooked up with the autonomic nervous system. So we'll study it a little bit, but not in any more detail than we really need to. We're also going to look at the ventricular system and cerebral spinal fluid in quite a bit of detail. And then we'll be able to leave that behind. That'll just be something that we don't exactly really build on, but we definitely will use it clinically. We also look at the blood supply of the brain. I think I start that most of it's next week in the second lecture, but we do get into it a little bit tonight, I think. And then we look at what they have called general principles. The author of the book has called it general principles. And one is this combination of contralateral and ipsilateral motor control. Contralateral means crossed and ipsilateral means same side. And I'll just leave it at that for this point so I don't bog down on this slide. But we're going to look quite a bit at that in this chapter. And then that concept should be useful to you as we study individual parts of the nervous system, like when we get to particular cranial nerves, for example. Your general understanding of the principle of contralateral and ipsilateral motor control will help. We'll also look at bilateral motor control of speech production. Most of the muscles that we use for speech production are controlled by both sides of the brain, but not all. For example, the tongue is mostly controlled by the contralateral side. The lower part of the face is mostly controlled by the contralateral side of the cerebrum, whereas the pharyngeal area and laryngeal area and the velum as well, they're bilaterally controlled. So that's what is meant by bilateral motor control for speech production. Some of the speech muscles are bilateral, some are not. Unilateral language mechanisms is another general principle, and you already know quite a bit about that. You know that the dominant hemisphere is the one responsible for most of language processing, most of what we would call language processing. And also we'll look at the general principles of cortical organization. And we'll also take a look at the most commonly used imaging techniques. Now, I think I asked you to read the whole thing, and it's not very long. It's maybe 4 or 5 pages of imaging techniques and the technology that's used to study the brain clinically. I'm not talking especially about research, but clinically, although it is used for research as well. But they go into more detail than I think is necessary, because there are only several tests that are used with people who have neurologic damage or injury 95% of the time or more. And some of the tests they mention, really, I've never seen a patient who's had it. So I just leave it out. So you'll see it's in the reading, but it's not going to be in the lecture. And that would be in the 3rd lecture. All right. This is a book. This is an illustration from the book, from the chapter. And they divide the nervous system, the central nervous system, I should say, into these 7 parts. And they use a combination of terminologies that are frequently used. And so it's kind of like being bilingual. That's why they did it, and that's why I put it on this slide. Mostly, we're going to use the language that is, well, let me just go through them. Telencephalon. When we're talking about that part of the brain that's referred to as a telencephalon, I'll mostly say the cerebral hemispheres. That's what it is. Now, where that term telencephalon came from is really from embryology, from the pattern of development of the brain. In that embryological development of the brain, there are 5 parts. Let me just go through them quickly. Telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And they sort of go from top to bottom of the brain. Essentially, they do. And you already have some familiarity with them. So we'll once in a while use those terms, but mostly we will refer to them a little differently and more directly. The telencephalon is the cerebrum or cerebral hemispheres. The diencephalon, and that's the second of these 7 things that the book refers to, 7 major divisions. The diencephalon consists of the thalamus and the hypothalamus. And you've already had an introduction to that. Thalamus we're not going to do much with. Thalamus we will. The mesencephalon is the same as the midbrain. And that, well, they're pointing here, and yeah, that's a good depiction of it. You can see the blue area, telencephalon, or cerebral hemispheres, the diencephalon here, the midbrain here, or mesencephalon. Then the metencephalon, again, that's the embryological term, but in embryology, that part, the metencephalon, gives rise to 2 major structures. One is the pons, so here it's numbered 4, and one is the cerebellum, the part of the brain that does the coordinating of our movements. And here they number 5. Then the myelencephalon is the medulla. It is part of the brainstem, and it's the lowest part of the brainstem. You go just below the medulla, and you've dropped out of the skull, and that spinal cord, which is the 7th part of the CNS. That's not part of the brain. So the brain consists of telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. We'll mostly use the terminology that is that I've gone through there. Okay. All right, now this is a ventral view of the brainstem, showing the cranial nerves. And it does show all the cranial nerves except for the number 1, which is the olfactory nerve, the one we do our smelling with. I'll go through and point them out, but they're well labeled, so you probably spent a little time with this. And I do believe that you already know them, and I think you probably studied how they are connected to the brain, not just the nerve and what it does peripherally. Like, for example, the hypoglossal nerve innervates the muscles of the tongue, and I think you know that from 344 or your anatomy and physiology course. But you probably also know, you've seen it come out of the brain before. But this illustration does show that. It's a photograph of a real brain. And going through it, they're numbered from top to bottom. And again, there's no olfactory nerve here. It's kind of above where the picture starts. It's up in this area. But the first one that they show is the optic nerve, number 2, optic nerve. They show number 3, which is the ocular motor. And I'm going to show 3 nerves right now, and I'm going to skip 1, number 5. I'm going to show 3, ocular motor, 4, trochlear, and 6, abducens. Now, where is 6? Right here. The reason I'm lumping them into 3 is that they all control the movement of the eye. They hook up to the extraocular muscles that turn your eyes to the right or the left or up or down or whatever you do. And we will study them a little bit. They don't have anything directly to do with speech, but understanding how they are affected in neurologic injury and disease is very, very helpful in making our diagnoses. You can easily, for example, you can easily see when somebody has a brainstem lesion as opposed to a cerebral lesion through examination of extraocular movements. And when somebody has a problem with an extraocular movement, it is very apparent. Okay, so now we're going to come back to 5, which is the trigeminal nerve, and here it is. That's a really big nerve, and it does a lot of things and a lot in speech and swallowing. And so we will study that in quite a bit of detail. Same for the 7th, the facial nerve, right here. Here it is. Controls facial movement, and obviously, and some other things too, but mostly we think about it as the nerve that innervates the muscles of facial expression. The next one is the vestibulocochlear nerve, and that is right here. That's obviously a very important one in communication. It's responsible for hearing. Sorry for skipping that. Why did that happen? Okay, here we go, sorry. I have to be more careful with my mouse. And then glossopharyngeal nerve, number 9, right here. That is primarily a sensory nerve, almost exclusively a sensory nerve. And it's very important in, not so much in speech, but in swallowing. It carries the sensory information needed for swallowing from the entire throat area, the larynx and the pharynx. And so it's extremely important. The vagus nerve, number 10, located right here, is also very important for speech and swallowing. It does control the velum. It controls the intrinsic laryngeal muscles, and it controls all of the pharyngeal muscles except for one little one that we never test anyway. So it's very important in understanding swallowing and also speech because of voicing. The accessory nerve, number 11, is shown down here someplace. Where is number 11? Right here. Okay. And we will study that briefly. It has to do with shoulder movement and head turning. And then the hypoglossal nerve, number 12, over here, is pretty important for us because it controls tongue movement. It's a motor nerve. Some of these nerves, by the way, and we'll go into this a little bit more in this particular third of the chapter, some of them have motor functions only, like 4 and 6, the eye moving ones, and 12, also the tongue moving one. Only motor, that's all they do. And some of them are what's called mixed modality nerves, meaning the nerve itself does carry sensory fibers or afferent fibers, and as well as motor fibers. All right, let's go to the next slide. Make sure I'm going to the next one. Yeah, okay. So elaborating on that point I just made about the modalities, we do have 12 pairs of nerves that innervate structures of the head and some in the neck, and those are the cranial nerves. They all connect to the brain stem, except for number 1 connects directly to the cortex, to the limbic area of the cortex, as a matter of fact. So of those 12 that connect to the brain, most of them are mixed modality, or I should say some. I don't. It is most, but some of them are mixed modality and some are single modality, and we will go into the specifics of those. We're kind of glossing it over to this time, but when we get to the actual cranial nerves, we'll go into a lot of detail. Not all of them, but the ones that are relevant for speech and cognition, speech and swallowing. The modalities that are served by the cranial nerves, and I'm just going to give you the language right now and a little bit of understanding, but it will kind of sink in over a number of lectures. You see that I've divided the modalities, or I didn't do it. It was done before I came along. The modalities are divided into the afferent components and efferent components. Afferent components are those that would carry information back to the CNS. Efferent components carry information out from the CNS to effector organs. We've used that term before, muscles and glands, and we're mostly going to be interested in muscles. Okay, so we've got that division, afferent and efferent. Now let's look at the afferent. There is this thing called general afferent, is I think what you usually think of when you think of sensation. It's touch, it's sense of movement. It is a sense of pain. That's all. Pain, touch, movement, that's all general afferent. Visceral afferent it's also afferent. These neurons carry information back to the CNS, but they're carrying it from different areas, mainly from the internal organs. In order to have digestion going on, for example, your brain needs feedback on what's going on down there. It's going to send information down to the stomach and down to the intestines to do certain movements with smooth muscles in a certain sequence, but it needs feedback as well. So the visceral afferent neurons provide that feedback as to what's going on with these structures that are involved in what we term vegetative functions. Then there's special afferent, and special afferent is hearing and vestibular function, and yeah, that's it. Hearing and vestibular function. I'm leaving off one. Oh, vision. All right, now we go to the efferent components. There's somatic efferent. Those are neurons that go just out to muscles, what you think of as the muscles of your body. There is a special category of muscle, it's the same kind of muscle fiber. If you look at it under a microscope, they're striated muscles. Most of the muscles of your body are striated muscles, except for the smooth muscles of the internal organs and the glands. So somatic efferent, these go all over your body. Branchial efferent neurons innervate muscles that are striated, but they're in a certain region of the body that was in embryological development, developed out of the branchial arches, something called the branchial arches. Now, I don't know if you studied embryology at all in your anatomy and physiology class, or perhaps some other class, but the branchial arches, as this embryo was developing, basically gave rise to the face and the throat, and the larynx, for example. The face, the throat, and the larynx. So obviously structures are involved with us. And the neurons that innervate those muscles are functionally, they're the same, but if you go back to the brainstem where they originate from, they're in kind of a column of cells that are aligned. The somatic efferent column of cells that give rise to those neurons are aligned. We'll look in a great deal more detail there. So the origin of the distinction comes from embryology, but anatomically there really is a distinction. When you look at the brainstem, all the branchial efferent neurons come from an area of the brainstem that is in a line. The somatic efferent come from a different line. All right. And then the visceral efferent neurons are neurons that go to the smooth muscles, the muscles that make up glands, and the muscles of the internal organs. All right, so now we're going to be using those terminologies a lot, the GA, the BA, or I'll probably say it out. I'm not going to give the letters, but those are the abbreviations. And sometimes in the slides you'll use the abbreviations. They're very common. I didn't make them up. All right, now when we go to the same sort of information, but pertaining to the spinal nerves, again, there are 31 pairs of spinal nerves, as you know. They're all mixed modality. When the cranial nerves, some of them are mixed, some of them are single modality. Spinal, they're all mixed modality. And again, we have the efferent and the afferent components because our spinal nerves are carrying information out from the central nervous system to give commands to effector organs, and they're getting information back to the central nervous system for sensation, etc. There's a distinction between somatic efferent. Now you know somatic efferent. Those are neurons that are going to go to striated muscles. And there are visceral efferent. Those are neurons that are going to go to smooth muscles, mainly for digestion. And those are found only from T1 through T12. So when we look at a cross section of the spinal cord, we'll look at that again. You'll see T1 through T12 is a little bit different internally, inside the spinal cord than the other levels of the spinal cord. All right, now looking at the afferent components, there's somatic afferent and there's visceral afferent. Again, you find the visceral afferent only T1 through T12. And just as a reminder, T1 through T12 would be those 12 thoracic level spinal nerves. That's what we're going to refer to as T1 and T12. T1 to T12, it's 12 of them. All right. Oh, and here we have a cross section through through the spinal cord with a spinal nerve, one spinal nerve, and its dorsal root and its ventral root. Now we would see this same pattern for every single one of the 31 pairs of spinal nerves. There's a nerve and it has two roots connecting it to the spinal cord. What's called the dorsal root has only afferent neurons. What's called the ventral root has only efferent neurons. So let me make sure I'm not missing anyone. So we have spinal nerves, our mixed modality. We've got the 31 pairs, afferent through the dorsal root into the dorsal or posterior horn. Let me point that out. In the spinal cord, and I think we looked at this in chapter 2, there is a dorsal root and a ventral root. And what this whole area is also referred to the spinal gray. It's kind of the H-shaped structure. That looks actually sort of gray if you look at a real section, and the areas around it in this dorsal band of tissue looks white. It's myelinated fibers. There are no cell bodies. And therefore there are no synapses. All the synapses, all the cell bodies and synapses that occur in the spinal cord take place in its H-shaped spinal gray matter. Some of them in the dorsal root, some of them in the ventral root. And so that's what this third bullet is getting into, is afferent through the dorsal root and into the dorsal or posterior horn. And you see here some schematic synapses occurring. Efferent neurons go through the ventral root, and they have their cell body located in the ventral horn. So let me make sure that's making sense to you. So we studied the anatomy of a single neuron, and mostly we have multipolar neurons. So this would be an example of a multipolar neuron. Notice that they didn't draw the details out. This is a schematic representation of a cell body, and this is an axon. And comes down here to a muscle, and there's actually a bouton there, connects to the muscle and transmits the electrochemical impulse across a cleft and makes that muscle contract. So here is something we're going to see again and again and again as we study it. It's sort of a very simplified representation of a neuron with its cell body, bouton, another cell body, axon, bouton. We know if we could magnify this and look in greater detail, that actually this cell body would have a number of dendrites and probably have many synapses form on it. All right, just flushing it out a little bit here, and I think we've covered the third point. Now the fourth point is about the autonomic nervous system, and that is shown here. Now they don't show any neurons, but this is the lateral horn. That is where there are cell bodies associated with autonomic nervous system function. That would be for digestion and things like that, where smooth muscles are involved. And you find that only from T1 to T12. All those connections, sort of occur around here and make everything in your abdominal and chest cavity function. Let's see. Okay, spinal nerves leave the spinal column through the intervertebral foramina. And that is not shown here. So I made the point, but now we're going to look in another slide and get into the details of that. I just wanted to point out here, foramina is the plural form of the word foramen. Foramen basically means a passageway through a structure. So your skull has quite a few foramina, as you know, and you studied them in 344. All right, now looking in a little more detail at that, I said in the last slide, let's go back, the spinal nerves leave the spinal column, the bony spinal column, the column of vertebrae. They leave that column through the intervertebral foramina. Now we see it. Here is one intervertebral foramen. So what we see here is two vertebrae, 1, 2. We see the disc. You all have heard of spinal discs, which sometimes herniate and cause problems from injuries or just also from aging. And so we got two vertebrae. The orientation is this. This would be the ventral side. The ventral side. This is dorsal. This is called the spinous process. You learn these things. You know that most of the weight-bearing functioning, the weight-bearing of the spinal column is borne by the spinal bodies and the discs. Here's where you carry your weight. The spinal cord itself is kind of pretty well protected, and obviously it should be because it's very delicate and very, very important. And it travels in this thing, which is called a vertebral foramen. It's not labeled, and sadly, it's not labeled. Now I got this illustration from someplace, and I thought it was so nice and clear, but that's the one thing I don't like about it. It doesn't have a label. It's the vertebral foramen. And those line up. So if you stack all your vertebrae up, those holes align and the cord is inside of it. And that provides it with a lot of protection. The only place where it's really open is right here. That's the intervertebral foramen. And what comes out of there is a spinal nerve, one on one side, one on the other. You can run into trouble even without a significant injury, just from the breakdown of the discs. The discs can break down here. Now here's the grayish colored disc, and that can herniate out right against the cord itself, or it can herniate out here next to the intervertebral foramen and cause a problem, a disruption, only with that particular spinal nerve on that side. So just depending exactly how that disc is herniating and where will set up the symptomatology and the signs and symptoms. Let me just throw a hypothetical example at you. Let's say it's a very severe herniation and it's halfway down the body. And it essentially cuts off, it squeezes it so hard, it cuts off the cord, so that below the midpoint of the body, that cord is not carrying anything up or down. It can't get past that point. And that does happen, by the way. It also does happen in spinal injuries where there's a fracture of a vertebrae that in functionally or actually cuts that cord in two. In that kind of a situation, you would lose all the functions for both sensation and for movement below the point of the injury. So let's just summarize it by saying you'd lose your lower extremities. We could be far more detailed than that, and we'll get into a map that shows that in a little while. But you'd lose everything below, all functioning below the lesion. Whereas if you had, if you had an injury, if you had a disc problem that only pressed up against a particular spinal nerve, you would lose whatever muscles that nerve goes to and whatever sensory zone it carries information back from. All right, so just starting to get into a little bit of clinical understanding of localization of function in terms of spinal cord this time. All right. Now, this will show this in a different way. Here we have the spinous process. First of all, I should say, it's a transverse section through a spinal cord. And here is the spinal cord. And you notice that it's floating in some liquid. Well, now maybe you don't know that, but the black thing is some liquid it's floating in, and we'll get into that. We'll get into that later. And it does have, this is the spinous, which we know is dorsal. So here we see a dorsal root, a dorsal root and a ventral root. I don't see a lateral root, so I guess this particular example is not taken from T1 through T12. It's either higher or lower. Here we see a vertebral body, and we see the disc. And then we see a dorsal root and a spinal root. And we see that they do come together and form a single spinal nerve. All right. And then I'm trying to focus on the clinical differentiation of a diagnosis of a disc herniation, which impinges on one or the other of these two possibilities. One in which the spinal nerve is impinged upon as it passes through the intervertebral foramen. And in that case, what would be lost would be the functioning of whatever that nerve does on that side. Actually, a neurologist could test the muscle functioning and the sensory zones and tell you in about 30 seconds where the disc problem is. If, on the other hand, this disc herniates directly against that cord and does it with such great pressure that it impedes the functioning of the cord below that level. So it basically nothing can get from the top down below the level, and nothing can get from the bottom up. All the afferent from down below can't get up. So you'd lose, in that case, you'd lose the functioning of all the muscles and all sensation, both sides of the body from that point and below. So we're just starting to get into a localization of lesions, and you can see the pattern, hopefully, that this is a fairly simple one. Once we get into the cranial nerves, they're a little more complex, but the principles are the same. So that's why I think it's very useful to start with the spinal cord, because once you kind of figure out how that works, it really works. Okay, let's look at more clinical implications of spinal cord injury. And this time, my focus is not on peripheral nerve versus central. It's on a cord injury and focusing on what happens differently depending how high up in the cord the injury is. Let me take an example that's not on here. Let's say, and I'll just refer to a very simple, conceptually simple kind of injury, a transection. It's just cut. Something happened. It could be a gunshot. It could be a knife. It could be a break in the vertebra that results in a fracture that cuts that cord. So let's put it up at C1. That's the highest level. What that individual will lose is everything in their body, except the head. So they would have all their cranial nerve functions. And that means if they are put on a ventilator, they can survive. And they could even speak, because you speak basically with your cranial nerves. So that's just an illustration of what happens if it's really high in the spinal cord. But now let's go down a little lower and talk about it in relationship to inhalation for both survival and for speech production. You know this term tidal volume, that is from your anatomy and physiology course, I think. Tidal volume is what you just breathe in and out. Now your lungs can breathe in someplace between 3 and 5 liters as an adult. That's the total capacity of your lungs. But you don't normally, take that kind of a breath. It's a much more relaxed breath. And it's at the typical adult, it's around 500 to 750 cc's. That's less than a liter. That's a half or a little more than a liter. And you do most of that work with your diaphragm. That's all you're doing, is you're just sitting there right now listening to me. Your diaphragm's working, but not much else is going on. You're not really using your abs. You could if you want to, but you don't have to. And so it's mostly the diaphragm, and that's enough to keep you alive, basically, and enough to help you. You can speak on diaphragmatic breathing too. So it's about the diaphragm. What nerve innervates the diaphragm? It's called the phrenic nerve, and it has 3 roots: C3, 4, and 5. Those are cervical level 3, 4, and 5. Those roots come together, form a single nerve. On one side goes to that half of your diaphragm. On the other side goes to that half of your diaphragm. And so what happens if the transection of the cord is above C3 is you totally lose your diaphragm. You can't breathe in. And in fact, for all intents and purposes, that person would function the same as if it's at C1. We already talked about C1 transection, so bring it down to C2. Let's go down a little further now. And C3, they're going to lose it all. But if you go to C4, what happens is they have 1/3 of their diaphragmatic strength, meaning they probably can't inhale 750 cc's, but they probably can inhale 1/3 of that, 250. Or if their baseline's 500, 150 or 200, something like that. Can they survive? Yes. What would they have to do? Breathe faster. They'd have to breathe faster. Could they speak? Yes, they could speak. How would that affect their speaking? Well, they'll have shorter phrase lengths. As I'm talking here right now, I am talking on more than just my tidal volume. I'm taking a little bit bigger breaths, which means I'm getting into my inspiratory reserve, and I'm pushing at the end of the phrase, so that I don't know how long my phrases are, but probably I'm speaking on a breath 10 seconds or something like that. How am I doing it? Taking a bigger breath and pushing a lot out. And for that, you need other muscles. But for inspiratory reserve, you need mostly your upper thoracic accessory muscles and nerves around here. So if you had a spinal cord injury that's down around C6, your inspiratory reserve would be okay. But your expiratory reserve, which I'm going to illustrate using right now, I'm going to talk until I'm almost out of breath and I'm using my expiratory reserve, and I can definitely feel I'm using my abs. What nerves are controlling those abs? They're down around here. They're down around the lower thoracic area. There's kind of a correspondence, so you just kind of go out from the cord. That's in general, those are the muscles that were innervated by the spinal nerves at that level. So you would lose expiratory reserve if you have a transection that's around, let's say T1, 2, 3, 4. But you would be able to speak. You'd have shorter phrases. You'd take more breaths. And the other thing that you'd lose if you had something that's, let's say, halfway down the cord, is you would lose some of your emphasis and loudness, because the way we emphasize things or the way we speak loudly is we push with the abs. And you would lose some of that kind of effect in your speech. All right, so I think we've covered everything under inhalation and exhalation, and we've talked about the importance of cord injuries at C3 versus 4 versus 5 versus 6. If you have an injury just below C6, you definitely can speak very functionally, but you do lose phrase length and you do lose emphasis. All right, now we're going to the next slide here, which shows this little map of the human body, which is referred to as a dermatome map. A dermatome is an area of the body that is innervated for sensation, and I should say general sensation. This is about touch, innervated by a particular nerve. And you can see the pattern here. Let's just start here. Let's get out of the head for a moment. But you can see that C3 provides general sensation for basically the front of your neck. C4 down around your upper chest. C5, 6, 7, and 8 down your hand. And you can see these are lines. They're topical lines. They're obviously imaginary lines, but a neurologist could, by testing the patient's ability to feel a light tactile sensation, discover, for example, that the person can feel here, they can feel here, they can feel here, but they can't feel here in C4 on the right. That's the right side of that person's body. And they would know that they had a lesion. They wouldn't know what type of lesion, but they'd know they had a lesion that affects either the spinal nerve or the dorsal root of C4 on the right. So the whole body is divided up like that. These are called dermatomes. And when we get into our cranial nerves, we'll look at dermatomes up there too. They are not shown very well here, but we'll divide this whole area up into dermatomes. So now you know the concept of the dermatome. You know how it could go into a localization of lesion. Let's see. So this slide is about clinical differential diagnosis of spinal cord injury versus spinal nerve injury at the different levels. I think we've kind of already gone through this. But we can relate it to the dermatomes now. So let's do that. Let us say that a spinal cord is transected, cut across completely at C1. What will be lost for sensation would be all these dermatomes up here. But hang on just one moment. Actually, I can't, but it's not much longer. What will be lost is these sensations for all those areas below C1 and below. And also muscles, but we're not talking about muscles now. We're talking about sensation and dermatomes. So I'll just leave it at that. If the transections at C6, they would have intact sensation here, and on the opposite side too, here down the arm, but not here. And the neurologist would be able to discover that they don't have the C6 dermatome, and they would lose all of these. And T6, the same thing. So all these dermatomes up here would function on both sides, but below T6, they would not feel anything. Now let's look at the example of a herniated disc interrupting ascending and descending pathways at C2 on the right. Okay, now, this is like a partial transection. The herniated disc is putting pressure on the whole cord, not just a nerve, but only on one side. That's usually not clean. It's not like 100% like a cut. You can cut into a cord halfway across, and that's 100% clean. You lose everything below it, and you don't lose anything above it, and you don't lose anything on the opposite side. But this is a herniated disc, so not be quite that clean, but nevertheless the pattern would be there. If the injury was on in the cord C2 on the right, what would be, and that's this side, what they would lose would be the dermatomes below C2 on the right, but not the left. If it was the whole cord transected at C2, they'd lose the dermatomes below it on both sides. And T6, the pattern is the same. We just go down a little bit lower. The herniated disc is T6 at the level of the T6 on the left, and it is impeding the flow of information up that half of the cord. They will lose all the dermatomes below T6 on the left. All right. Herniated disc interrupting the L3 spinal nerve on the left. So we come down here to L3 and go to the left. Here's what they're going to lose. This on the left. Okay. All right. And a mandibular division of CN5. Well, we can't see that very well here, but do you remember? I think you know this from your anatomy and physiology nerve class. I mean, remember the trigeminal nerve provides sensation for the face, and it's divided into 3 divisions: the ophthalmic for the forehead, the maxillary for the upper cheek area, and the mandibular for this area. And so if the mandibular division of cranial nerve 5 on the right, if there's a herniated disc involving that one, well, first of all, let's go here. That can't happen because there are no discs up there. This is brainstem. So I don't know if I put that in as a trick question, trick item or what. You certainly can have a problem with the mandibular division of cranial nerve 5 on the right. Like, for example, a little tumor, or more likely some kind of an injury that injures that as it leaves the skull, and then you would lose the sensation for the lower jaw area on the right. It just wouldn't be from a disc problem because we don't have any discs in our head. We only have them in our spine. All right, 2 more slides and we will have it. This is going to, of the slides that we're going through right now, these are going to make the least amount of sense, but I'm going to introduce them anyway. And you can see that they're not from the book. But they're extremely clear, and you're going to see this again, this exact slide again and more than once as we work our way through the cranial nerves. But I just want to relate what we've studied so far today to the brainstem and to the cranial nerves, because that's the guts of what we're really learning about here, is those cranial nerves and how they function, in addition to the cerebrum, of course. And so here are the cranial nerve names. You can see them. And what I've shown here is which ones have which modalities. Remember modalities earlier? Somatic efferent. Somatic efferent neurons are going to go to muscles. And they're going to go to muscles that didn't grow from branchial arches. Up in this area, it happens to be 4 muscles, 4 sets of muscles. The extraocular moving muscles, that's 3 of them, 3 sets, ocular, motor, trochlear, abducens, and the tongue. Excuse me, the tongue. And so what I want to do is, first of all, explain the concept of a nucleus again, because you've heard the term nucleus even in this course when we said that a cell body has all the organelles of any cell, including a nucleus. That's a cellular nucleus, the little part of a single cell. When the rest of the time in the course, when we use the word nucleus, and we'll use it a lot, that nucleus is defined as a collection of cell bodies within the CNS, a collection of cell bodies within the CNS. And anytime you have a cell body in the CNS, there is information processing going on. In other words, it's not just simply passing an action potential from one neuron to the next, like a relay race. There's a whole bunch of dendrites and a whole bunch of synapses, and information's getting processed, and something out of the message coming out of it is a little bit different than what it went in. So we're going to talk about nuclei, those nuclei, a lot, and they are there are 4 of them in the brainstem that are somatic efferent, and I'm going to point them out, and then I'm simply going to point out that they're aligned. That's all. So that's right now we're kind of look at these nuclei in one way, and then we'll come back and look in a lot more detail. Some more than others, because the ones that are most relevant to speech we'll look at the most, of course. So let's look at the ocular motor first. There it is. And then the next one, trochlear, is located right there. And then the next one, abducens, is located right here. And notice that they are, well, they colored them the same, try to make it easier for us. And they're more or less aligned in the brainstem. They're kind of close to the dorsal. They're not scattered all over. Like this one is not aligned with those other 3. This one is not aligned with those other 3. These are aligned. So in the development of the brain, they sort of came from the same origin. And then the hypoglossal nucleus, responsible for tongue movement, is down here in the medulla. I should point this out as we're sitting here looking at this, that we're looking at a side view. It's not exactly a sagittal section, but we're looking at a side view. There's a kind of a 3-dimensionality to it, which is why it's not just a sagittal section. So we're looking at the side, and here are the 3 different parts that we can see here: a midbrain, a pons. We can also see a little bit of the cerebellum, and a medulla. And at some point, we can't see exactly where, but right around here, where the hypoglossal nucleus ends, the CNS drops out of the skull, and down here would be spinal cord. Okay, so we've got these 4 nuclei. We know what somatic efferent means. We know all those neurons are going to innervate muscles. They're all striated. They all grew from what's called somites, not from branchial arches. And then we get here to these 3 nuclei: the trigeminal motor nucleus, the facial, and the nucleus ambiguus. They're all concerned with speech production and or swallowing, or both. And I'm going to point them out. Trigeminal nucleus, right here. Trigeminal motor nucleus, dark blue. And then the facial nucleus, more or less aligned. And then the hypoglossal, the nucleus ambiguus, right here. Let me make sure the line's going to it. Yes, it is, nucleus ambiguus. And that nucleus ambiguous contains, again, just going over again what a nucleus is, if not an organelle in a cell, it's a collection of cell bodies in the CNS. And so these cell bodies they make up the nucleus. are sending their axons out. They're sending their axons out, and they're sending them all out to muscles involved in speech and or swallowing, to the velum, to the pharynx, and into the intrinsic larynx. Okay, so we're going to look a lot at these. Visceral efferent, those nuclei are sending axons out of the CNS2 to effector organs, but this time to glands or to smooth muscles. Edinger-Westphal is a nucleus that innervates the part of the lens of the eye. So we're not going to look at that at all, actually. I mean, we can label it. Here it is, Edinger-Westphal. Superior salivatory nucleus, where is that? Superior salivatory nucleus right here, and inferior salivatory nucleus right there. They send messages out to your saliva glands to produce saliva. And we have an awareness of that, but we'll never, as SLPs, will never be involved in putting together part of our clinical picture for our patients based upon what we see going on with saliva. And then the dorsal vagal nucleus, located here, it's kind of gray here, is a really long nucleus occupying the length of the medulla that sends neurons mostly down, not only down, but mostly down to internal organs of the body to tell your muscle to do what, your stomach to do what it does, and your intestines to do what they do, etc. Okay, one more slide and then we stop. This is a slide showing the afferent nuclei. So, and we have somatic afferent nuclei involved with sensation, special sensory, and that's auditory and vestibular, and then visceral afferent nucleus solitarius. And we're almost there. You can see it is the same slide. It's the same picture, but we're looking at different parts of the picture now. Let's start up here. This is the trigeminal nerve. It exits from the side of the pons, and it does have some motor fibers in it, and we're not going to talk about those now, but we're going to just focus for this slide on the sensory part. And the sensation would come from the entire face, and even the top of the head, the entire face, through those three divisions: the ophthalmic, the maxillary, the mandibular. You can see them labeled here: ophthalmic, maxillary, mandibular. And not only that, but also the entire oral cavity, top and bottom, the whole thing, right back to the port called the oral pharyngeal port. If you look back and you see the faucial arches, the border of the velum, behind that is outside the range of this nerve, but in front of it is inside. So it's the face, the top of the head, and the whole mouth, but not the back of the mouth. And there are three parts to this very, very long nucleus associated with somatic afferent functions. This part, I'll give them a name in a moment, but it's all the way up into the midbrain. This part, which is in the pons, and this part, which goes all the way down, actually, well, it goes all the way through the medulla, and then it links up with a similarly functioning column of cells in the spinal cord. Right now, we're just going to give them names and functions. The part that goes up toward the midbrain is the mesencephalic trigeminal nucleus, and its job is to subserve proprioception and kinesthesia. You can imagine that's pretty important for speech production and functioning up. You kind of have to know what your face is doing and what's going on in your mouth, where your tongue is, what it's touching, you know. The principal trigeminal nucleus, which is located here, is responsible for fine touch. Proprioception is a perception of position. Kinesthesia is perception of movement. And fine touch is perception of light touch. And then the part that goes down into the medulla is the spinal trigeminal nucleus, and that's responsible for the processing of pain and temperature information. Okay, special sensory nuclei are responsible for hearing and vestibular function for balance. And I think they're shown here. I'm just looking around for them. We will study them a little bit. Actually, am I missing? Am I missing? Yeah, they don't have, I have it right here, not shown. Okay, so we're not gonna look at the location of those, but they're right around here. And then the visceral afferent nucleus, where the two of them right and the left, it's called the solitary nucleus or the nucleus solitarius. Either one is fine. And that is located right here. Now, it doesn't show it very clearly, but it's a long nucleus. It goes the length. It's this little white one, but it does go the length of the medulla. And that is responsible for processing all afferent information from the throat and laryngeal area. It is very, very key to swallowing. So we will look in detail at that. So that is the part 1 lecture from chapter 3. We'll go into slides number 15 through 29 in number 2 next week.